Thank you so much for being here today. So zooplankton are broadly defined as all of the animals that live in the aquatic environment that cannot actively swim against a current. So they range in size from microscopic animals not visible to the naked eye, all the way up to macroscopic animals, including jellyfishes and salps. Mesozooplankton are those zooplankton that fall within the 0.2 to 20 millimeter size range, and they are the primary group of zooplankton as far as size distribution that I work with in the York River. And zooplankton are important for a variety of reasons, the first of which is that they form a critical intermediate link in all aquatic food webs by grazing on phytoplankton and then in turn being eaten by a variety of larger organisms. They're linking the primary producers within the aquatic food web to the higher trophic levels. And then additionally, they play a really important role in the biogeochemical cycling through something called the biological pump, which is the suite of biological processes whereby surface community production, i.e. fixed carbon, is transported down to depth. And so zooplankton contribute to this by creating fecal pellets, or poop, which passively drift down into deep waters and support deep ocean food webs, including microbial communities. But they can also actively transport this production to depth through diel vertical migration. That's a process whereby they migrate up to the surface waters at night to feed under the cover of darkness, and then they're migrating back down into the deep waters during the day to avoid predators. When they're at those deeper depths, they're digesting their food and releasing these fecal pellets in the depths themselves. What is not consumed or remineralized in the water column is eventually deposited onto the seafloor, where it can be buried and effectively sequestered from the environment. And on a global scale, only about 0.1% of the surface carbon is buried, and this is on the order of millennia, but it's a significant process that aids in carbon sequestration, which also aids in the regulation of our carbon and our climate cycles. So zooplankton, like this copepod here on the left-hand side, they ingest particulate organic carbon in the form of phytoplankton, so they're grazing on phytoplankton. And they release dissolved organic carbon through sloppy feeding, but also the excretion of waste. But egestion is the process that leads to the development of these fecal pellets, which are dense little compactions of particulate organic carbon. And because these sink, they play an important role in that export of organic carbon. And on a global basis, zooplankton ingests between 9 to 17 gigatons of carbon per year. And then in turn, they're producing fecal pellets that contain roughly 6 to 10 gigatons of carbon per year. So thus, if just 0.1% of that carbon is eventually buried in the sediments, that's roughly 6 million tons of carbon that are effectively sequestered per year. And this kind of reiterates the importance that zooplankton and their fecal pellets play in the global carbon cycle. But not all fecal pellets are created equal. So different species or taxonomic groups of zooplankton produce very morphologically distinct fecal pellets, which impacts not only their carbon content, but also their sinking rates, and thus also the likelihood of them being remineralized in the water column versus buried in the seafloor. So for example, while all fecal pellets sink fairly quickly, salps, which are a gelatinous zooplankton, they create really dense fecal pellets that sink up to 10 times faster than copepod fecal pellets. And for this reason, salps are commonly called the export express of open ocean ecosystems. So just with this one small example, you can see that the structure of the zooplankton community itself can affect how much of the carbon is sinking out of the surface waters. And this variation in the zooplankton community structure leads to different rates of export around the world. So pictured here on this slide are fecal pellets that were collected with sediment traps in a variety of sites around the world. The Southern Ocean is dominated by those long cylindrical krill fecal pellets, whereas in the Sargasso Sea, amount of pellet flux, it's less productive than some of these other regions that are pictured on the map. However, you do see in that second photo, the one that says C, that export can peak during salt blooms due to just like the sheer density of their pellets. And this information is really critical, all of this to collect for understanding global carbon flux and export hotspots. And while the scientific community has done quite a bit of analysis of fecal pellet export and production at marine sites, there's been very little research done on fecal pellet production in estuarine ecosystems like the Chesapeake Bay, and never on the community level. And so this is the motivation that's really underlying my research here in my thesis work at VIMS. The major research question that I'm aiming to answer in my thesis work is, how do diel and seasonal changes in zooplankton community structure affect the fecal pellet production in the York River? And my hypotheses include that we would see more and larger zooplankton at night in the surface waters due to that migration of larger animals into the surface at night to feed, which would subsequently lead to increased production of fecal pellets, and also larger fecal pellets because there's that allometric relationship between the animal size and their fecal pellet size. I expect to see some biomass peaks in the zooplankton that lag slightly behind, but kind of follow the same cycle of our phytoplankton biomass peaks. So roughly in the spring and the late summer are when we see our phytoplankton blooms in the Chesapeake Bay. So I expect the zooplankton biomass to kind of lag behind, but peak after that, which would subsequently result in fecal pellet production peaks as well, because there's more animals in the surface creating the pellets. And then I also expect that gelatinous zooplankton exert a top-down control on fecal pellet production. And the reason for that is that copepods are typically the most abundant animals in most aquatic ecosystems, but definitely also in the York River. And so those are the animals that I expect to contribute the most to the fecal pellet production that I'm able to observe. And there are organisms called comb jellies or ctenophores, and they predate on the copepods, and they create mucus masses, and then in turn they're actually eaten by chrysora or banettles. And so these chrysora, if they're grazing down the amount, or well, not grazing, if they're eating the comb jellies, who are then eating the copepods, if there's a lot of banettles, that may mean that there's more predation on those comb jellies and less predation in turn on the copepods. So more copepods, more fecal pellet production. So these gelatinous zooplankton, their presence or absence can affect and change the dynamic of the food web by removing predators and prey. So my study site is the York River, right here, of which I sampled both a mesohaline and polyhaline region. And so the mesohaline region is up here near the Claybank area, and the polyhaline region is down here by Goodwin Island. There were 4 reasons for this choice. The major one is that I wanted better spatial coverage and accounting for possible variability in the community structure or makeup due to salinity differences. Each location was within a 20-minute boat ride-ish from the VIMS boat basin, so this made it logistically efficient and minimized the amount of time between collecting the animals in the field and then subsequently using them in my experiments. There were permanent and lighted stationary markers at each of the locations, which allowed for repeated sampling at the same location throughout the course of the year, and also easier navigation to the sites at night. And then finally, there is supplementary environmental data available for each of these locations from the Chesapeake Bay program, which I'm hoping can help enrich my analytical work at the end of my, my lab analysis. I did collect all the samples that I used in my project. I went out roughly once per month in the polyhaline region and roughly once per major season in the mesohaline region over the course of a little more than a year, so August 2019 to November of 2020. And the first net that I would pull during each of these sampling events was a 1-meter ring net with 200 micron mesh that was also outfitted with a flow meter to calculate the volume of water that was being filtered through the net during the collection. And then once I brought the cod end, which collected the zooplankton sample, on board, it was split immediately with a fulsome plankton splitter, and 1/2 was preserved in 4% buffered formalin to later be used for taxonomic analysis. What I'm planning to do is pass the preserved samples through a 2-millimeter mesh sieve so that I can separate them into 2 distinct size classes, of which the larger than 2-millimeter animals, I'm planning to enumerate and identify all of them in that portion. And then I'll be taking a subsample of the animals smaller than 2 millimeters to identify and enumerate using microscopy. And then the other half of the toe sample for this net was passed through 5 nested sieves of various mesh sizes. And these contents were then concentrated on Nitex filter discs, which were brought back to the lab to calculate and weigh the wet and dry weights of the organisms per cubic meter of water that was filtered for each size class. So in addition to this major quantified toe, I also did 3 additional zooplankton toes during each sampling using nets of 200, 500, and 1600 micron mesh to collect animals for my fecal pellet production experiments. And the animals, when they were brought up on board, they were immediately and very gently released into 5-gallon buckets of ambient seawater. So just collected right off the side of the boat at the research site, and they were then transported back to the boat basin where I set up my experiments. So in order to quantify the fecal pellet production by the entire zooplankton community, I performed sets of community-level fecal pellet production experiments. So this schematic on the left-hand side shows the methods for preparing these experiments. But essentially, subsamples from those large 5-gallon buckets were gently released inside stacked mesh bottom containers of various sizes in order to live size fractionate the animals into 5 distinct size classes, which were then placed into an outer closed container of whole unfiltered seawater that was also collected from the study site. And then they were incubated at in situ temperature and light conditions for the duration of the experiments, which was typically 4 to 6 hours. The zooplankton were contained in the inner container due to the mesh. And over the course of the experiment, they ate and produced these fecal pellets, which sank down through the mesh and into that outer container. I then was able to filter down the contents of the outer containers into centrifuge tubes, which were frozen until I was able to get around to analyzing them. And there were 2 replicates of each of the 5 size classes, as well as 2 controls for a total of 12 experimental units per community level experiment. And once the water in the outer containers is filtered down, the contents look a bit like this. So in this circle here, you can see the ellipsoid shape of a copepod fecal pellet, along with that's a centric diatom next to it for size reference. And in the second circle here, you can see a long cylindrical pellet, which is more representative of a euphausid or a mysid. And so essentially what I do is I filter that down, put it in a petri dish. I search through the contents and use a Pasteur pipette to gently separate the pellets from the other particulate organic material or non-organic material that's in the water. And then I release them into a separate dish of filtered seawater to isolate them. And then I use our microscope and the camera that's outfitted on the microscope to take photographs of each of the pellets, and use the analytical software that's on our camera system to measure the length and width of each of the pellets. And then these measurements are the ones that I use to calculate the biovolume of the pellets and the total biovolume of pellets present in each of the experimental units, so each of the containers. And then this biovolume information will be used to will be combined with future CHN analysis to develop a biovolume to carbon ratio, which can then be used to estimate the carbon content of pellets in the experiments where there may have been insufficient weight of pellets available to perform CHN analysis. And just to give you a little bit of extra perspective as I'm sharing my results, I wanted to give you just a basic idea of which types of animals are in each of these size classes. This is completely qualitative. I have not had the opportunity, unfortunately, to get around to my taxonomic identification yet, but just from my observations while setting up my experiments and analyzing my experiments, I can give you a little bit of an idea of which animals are in each of these size classes. In the smallest size class, we've got a variety of tiny little animals. This does shift based on the season. So ultimately, it's mostly dominated by copepods of various types. So we've got like 3 different types of copepods here. You can see their body shapes and plans are very, very different. We also have a lot of larval crustaceans. So we have larval crabs, larval barnacles, so Nauplii, a pretty significant amount of cladocerans, that are usually in the summer and fall. And then in the winter and spring, there were a lot of rotifers, which I forgot to put a photograph of them on here, so I apologize for that. But also in the summertime, we see an increased amount of diversity in the copepods. So I see a lot more different types of copepods in the summer versus in the fall, in the winter, in the spring. Also in the summer, I collected quite a few larval gastropods and bivalves, which was really exciting. And as you move up into the larger size classes, you get to other larval crustaceans, including crab megalope, and some of the larger Acarchatansa copepods. They can be quite large. They can be from that 0.2 millimeter size class all the way up to the 2 millimeter or more size class. And also polychaete worms, so some bristle worms. They also make up quite a bit of that size class. As you move up, we get some of those larger animals. So these are mysids, and these animals I've only collected at night, which is part of the reason why I'm doing the diel vertical migration study, is to collect some of these animals that only come to the surface at night. In the summertime, I found a lot of chaetognaths, so they're arrowworms, and they're not like an annual member of the zooplankton community. They do have a seasonal cycle, so they come in during the summer and fall. And then also I've been able to find quite a few larval shrimps. So those are more common in the summer as well. And also larval fishes. So we get quite a few of those in the summertime as well. And then that largest size class is mostly gelatinous zooplankton. Ctenophores, or those comb jellies. Enjoy this moment of zen of watching it refract the light down its combs. It's really beautiful. And of course, the dreaded bay nettle. So we have lots of those this past summer for sure. And I'll explain some of those gelatinous zooplankton trends that I found in the next slide. So on this slide, I'm showing the biomass results from my polyhaline sampling site, and they're depicted in this graph. So along the x-axis, you're going to see the year and also the month of each of the cruises. Where there are two bars next to each other on the same month, that means that that first bar is the daytime cruise and the second bar is the paired nighttime cruise. For months with just one bar, that means that we were only able to sample during the day. So those are all daytime cruises if there's just one bar. I did place an X on the months in which we were not able to go sampling due to weather or COVID or participation in the Antarctic cruise in the winter of 2020. And along the y-axis is the dry weight of zooplankton in grams per in milligrams per cubic meter of water filtered. And in addition to each of those 5 size classes that I mentioned, I also added a separate category for gelatinous zooplankton in order to allow a better visualization of those seasonal community shifts in the zooplankton community. And the term gelatinous zooplankton refers to ctenophores, so those comb jellies, the chrysora or bay nettles, and in one circumstance here in this August sampling, moon jellies. And so some of the trends that we see here, we do see an overall trend of higher biomass per cubic meter of water filtered at night versus during the day, with the exception of this November 2019 cruise. We see migration of intermediate size classes, especially the 0.5 to 1 and 2 to 5 millimeter size classes into the surface waters at night. And although we know that there are gelatinous zooplankton present in the York River in the winter and spring, the biomass in total of the gelatinous zooplankton peaks in the summer. And I also saw a pretty clear variability interannually between the summer of 2019 and the summer of 2020 as far as the gelatinous zooplankton biomass. So the nettles were very rare in 2019 in the summer, but in the summer of 2020, I was collecting dozens of the nettles in that July and August cruise frame. So there were lots and lots of them. And there's also pretty low biomass in April, which was counterintuitive to what I expected to find. I thought that that would be one of those peaks after the phytoplankton bloom. So now we can look at some biomass results from the mesohaline sampling site. Once again, the year and month are along the x-axis, while the y-axis is the biomass. And so because this sampling was much more sporadic, I only included the months in which we actually did go out along the x-axis. One important thing to note is that although this graph is roughly 1/3 scale along the y-axis of the polyhaline, in general, the biomass in the mesohaline zone is on a comparable scale to that in the polyhaline zone. It's just that one huge peak of biomass in August that sets that other graph on a completely separate scale. So here we still see a fairly consistent diel-vertical migration signal, with the exception of this August 2019 cruise. And unlike the polyhaline site, the nighttime tows in both August 2019 and in July of 2020 yielded Chrysaora, or the bay nettles. There were 4 in each of those 2 cases. However, it was interesting to note that in both of these tows where we did get Chrysaora, there were no ctenophores, whereas in the polyhaline site, I typically caught the ctenophores and Chrysaora together. So that's kind of interesting and something I'm going to look a little bit more into as I'm identifying samples. And so in addition to the biomass, I do have some preliminary results from my community level fecal pellet production experiments. So these are the fecal pellet production experiments that I've been able to analyze from the polyhaline York River site. So along the x-axis here are the dates of the cruises I've been able to finish analyzing, and on the y-axis we have the biovolumes of pellets, and these are average biovolumes. So basically I averaged the calculated pellet biovolume across the 2 replicates per size class, and then I subtracted out the average biovolume of the ambient pellets that were present in the 2 controls per set of experiments to present the data in this way. And once I perform CHN analysis, I'll have the fecal pellet carbon production for each size class. But for now, I just have the biovolume of pellets created. So a few things to note: the daytime pellet biovolume production was highest in March during the typical spring bloom, and lowest in February, which was prior to the spring bloom. November 2019, as far as the biomass data, was the one anomaly as far as the biomass of the animals being lower at night than during the day. And ironically, the pellet biovolume was actually much more during the nighttime than during the day. So I'm curious to take more of a look at that to see why that is. In February, the biovolume of ambient pellets in the controls was higher than the biovolume of pellets in the 3 largest size classes. So we really only have some positive, I guess, production in those smaller size classes. And just to let you know, I am planning to finish the analysis, of course, of these, the August, September, and November experiments. So these are just the daytime cruises that I've been able to analyze so far. When I return from this research cruise, and I'm hoping that these will provide me with more insight as to the relationship between the animal biomass and their pellet production, as those months had the highest biomass of animals in this site. And I'm hoping that the August experiments in particular will provide more insight into the impact of the gelatinous zooplankton on the production of pellets. And here are some preliminary results from my experiments that I was able to analyze from the mesohaline cruise. So once again, the graph is oriented in the same manner. A few interesting things to note: the November production showed a reverse relationship than in the polyhaline zones. There was lower production of pellets at night versus during the day. And that was kind of interesting. The biomass was much higher in the nighttime cruise. So I'm thinking that may be a result of the copepods and other animals actually moving around and breaking up the pellets and eating them as well. The February cruise had higher overall biovolume of pellets created relative to the November, which is consistent with a higher biomass of animals present in daytime February than in the daytime in November. And I do have a set of experiments from July 2020 as well as November 2020 that I've begun analyzing. I tried to get them done, but I didn't get them done in time, which will provide a more comprehensive seasonal perspective. And so in order to just quickly summarize, there is generally a clear diel-vertical migration signal throughout most of the year in both of the sites. We do see seasonal changes in the community, including most notably the gelatinous zooplankton community in the summer. Fecal pellet production seems to vary by size class. So I'm seeing so far that it seems to be dominated by those smaller size classes and the animals within them, and less production, relatively speaking, by the larger animals. And from here, I'm going to be continuing to analyze the remaining pellet production experiments, both community level, and also I did some taxa-specific experiments. Then I'll jump into taxonomic identification of preserved samples in order to get a better grasp on who's in what size class. And then eventually we'll be performing that CHN analysis of the pellets and the animal rate of fecal pellet production in each size class on an annual and diel cycle. So with that, I'd just like to acknowledge my advisor, Debbie Steinberg, my committee members, Mark Brush, Margie, and Carl Friedrichs, of course, my Steinberglers and all the helpers that have helped me with all of this, and my funding sources, the Commonwealth of Virginia, the Kelly Watson Fellowship, Beasley Fellowship, and of course, VIMS.